Adhesion G-protein coupled receptors. The nervous system is composed of different cells including neurons, glia, and their precursors. During development, neurons have to migrate from their origin to the proper destination, adopt diverse morphologies, and create specific dendritic and axonal contacts with target cells. In order to perform all these functions, neurons and glia need to have some type of communication with their neighboring cells and with the extracellular matrix surrounding them. The cells of the nervous system do it through adhesion G-protein coupled receptors. Adhesion GPCRs are the second largest class of G-protein coupled receptors. What makes adhesion GPCRs unique is that their extracellular domain is significantly longer than other classes of GPCRs. These long segments are often binding sites for adhesion proteins, and they also contain an autoproteolysis inducing domain, which is cleaved when bound to certain signals, activating the GPCR's intracellular domain. Therefore, adhesion GPCRs are involved in adhesion and signaling.
aGPCRs can be activated through a mechanical force-based mechanism. They can function as mechanoreceptors when sensing changes of high dynamic range that include the forces of shearing, bending, and vibrations. In the example shown, the aGPCR latrophilin has been found to sensitize mechanosensory neurons in Drosophila while responding to a probing stimulation. The removal of latrophilin causes a drop in the firing frequency of action potentials, demonstrating that latrophilin is a mechanoreceptor that sensitizes the neurons for mechanical stimulation. aGPCRs can also sense mechanical changes of low dynamic range. For example, aGPCRs can sense mechanical properties associated with the rigidity of the extracellular matrix of Schwann cells. Schwann cells can secrete a protein known as laminin 211 that helps to strengthen the extracellular matrix. Laminin 211 causes a stiffening of the ECM that causes the menoterminal fragment of the aGPCR to face some mechanical force. A specific sequence known as a stachel sequence may need to be exposed by the mechanical stimulation of the aGPCR to allow for receptor activation, causing an increase in levels of the second messenger, cyclic AMP.
The role of one aGPCR in a Schwann cell signaling pathway has been empirically determined. Schwann cells are responsible for creating the myelin sheath that encases axons, and the aGPCR, GPR126, is responsible for regulating this process. GPR126 binds and activates the alpha subunit of a stimulatory G protein. Once activated, this G protein activates the enzyme adenylyl cyclase, which increases the concentration of the second messenger cyclic AMP. Cyclic AMP activates the kinase pKa, which eventually causes the upregulation of genes required for both Schwann cell differentiation and myelination.
One of the first studies to define the essential function of GPR126 in peripheral nerve development and myelination in mammals was done by Monk et al. In 2011. In their paper, they compared a wild-type GPR126 mouse to a knockout GPR126 mouse. They showed that a mutation in GPR126 caused severe congenital hypomyelinating peripheral neuropathy in mice. This can be seen when you compare the wild-type picture on the left to the mutant picture on the right that lacks a fully developed nerve. They also showed that a mutation in GPR126 caused nerve and limb developmental defects, as well as reduced expression of several differentiated Schwann cell markers, which can be seen in the real-time PCR analysis. While SOX10 is present in both the plus-minus and minus-minus background, markers DHH, EGR2, and GPR126 itself are not detectable in the mutant nerve. Negative controls show no bend, and GAPDH acted as a positive loading control. The importance of this aGPCR is highlighted by the dysfunction caused by its mutation. In humans, GPR126 mutations have been known to cause a lethal condition characterized by limb malformation and reduced expression of myelin genes. Thus, future studies of aGPRs in the nervous system have important clinical implications.